Hi everyone, this video is on lactose operon, which is involved in the regulation of gene expression, particularly in prokaryotes. Now, what is this operon? Operon is a concept present only in the prokaryotes and not in eukaryotes. This is the first and foremost point which you have to remember. It's present only in the prokaryotes. So, what does bacteria do? It consists of different operons. Operons like, for example, lactose operon. It also has tryptophan operon. Yes, tryptophan operon. So in this video, we'll concentrate on lactose operon. So an operon is a package containing series of genes arranged sequentially, and these genes code for proteins involved in a particular metabolic pathway. Yes, for example, in lactose operon, these genes produce proteins that are involved in the lactose metabolism. Yes, so along with these proteins are arranged, these genes are arranged in sequentially along with other factors which are involved in the promotion of the transcription of these genes. Yes, so various operators and promoters along with these genes which are responsible for the lactose metabolism are arranged as a one operon model. So this is the operon model. Now let's get into the details of lactose operon. So initially what you have to know is, in a bacteria, it utilizes glucose as its fuel. Okay, so whenever glucose is present, it utilizes glucose for its energy. If glucose is not present, then it has to utilize lactose. So this is when this lactose operon come into play. So we have to understand what are all the conditions which stimulate this lactose operon and what are the conditions which will not stimulate the lactose operon. Now let's understand the lactose operon. So this is the operon which consists of 3 genes. So what are these 3 genes? These 3 genes are involved in producing proteins which are necessary for the lactose metabolism. So one is LAC Z gene, the next is LAC Y gene, the next is LAC A gene. So LAC Z gene codes for the enzyme beta galactosidase. The enzyme beta galactosidase, and LAC Y gene is necessary for the enzyme permease, and LAC A gene for the thiogalactoside transacetylase. Thiogalactoside transacetylase. Yes, so these are the 3 genes which are involved in the lactose metabolism. So whenever when a bacteria has to utilize the lactose, these 3 genes have to be transcribed. So for that, before these genes, what is present? The site present is the operator site. Operator site is present. And before that, promoter site is present. A cap site, which is the catabolite activator protein site, and then the LAC I gene. LAC I gene. Now this LAC I gene produces a repressor protein. LAC I gene produces a repressor protein. This repressor, if it goes and binds to the operator site, if the repressor goes and binds to the operator site, yes, what happens? The transcription cannot take place. So if the repressor goes and binds to the operator site, then RNA polymerase cannot undergo transcription. Yes, RNA polymerase cannot initiate transcription. So if the operator site has to be free for the RNA polymerase to act. Yes, so the LAC I gene produces a repressor protein. The repressor protein, if it binds to the operator site, then it is blocked. RNA polymerase activity is blocked and transcription is blocked. So the next fact which you have to remember is, you have the catabolite activator protein, right? So it is the catabolite activator protein. So this is the catabolite activator protein which can bind to cyclic AMP. So if catabolite activator protein binds to cyclic AMP, then this will bind to the cap site. So as it binds to the cap site, so when the cap site is bound by cap protein plus cyclic AMP together, then this will also favor the transcription. So what is a factor that favors transcription? One is the which favors transcription, or which favors the operon to operate, is the first one is the operator site should be empty. Yes, the operator site should be empty. And the second one is the cap site should be bound with cap protein plus cyclic AMP. Yes, so this will enhance the RNA polymerase to initiate transcription. Also, there is one more factor. If the lactose is present, if the metabolite is present, the lactose can produce a inducer molecule. Okay, so lactose can produce an inducer molecule called allolactose. This allolactose can bind with the repressor protein and it can inhibit the activity of the repressor. So if in the presence of inducer also, the transcription can take place. Okay, so first is the operator site should be empty, the cap site should be bound with the cap plus cyclic AMP, and then the inducer has to be present. If inducer is present, it can block the activity of the repressor. Now what are the factors which will inhibit, which will not favor transcription, or which will not favor the lactose operon or any operon? Is if the operator site is bound to a repressor, if the operator site has a repressor on it, and if the cap site is not bound to cyclic AMP and cap protein, if the cap site is not bound. Yes, so and in the absence of a inducer, the lactose operon will be inhibited, or it will not be functional. So these are the factors which can stimulate the transcription of the operon, or which can inhibit the transcription of the operon genes. So let's see in the three scenarios in which the lactose operon is off or on. So one is if the glucose is present and lactose is absent. So I had told you, whenever glucose is present, the bacteria will utilize the glucose only. So when glucose is present, what happens? Again, let us revise this. LAC I gene, after that is your cap site, after that is your promoter site, and then operator site, and then this is the LAC Z gene, LAC Y gene, and LAC A gene. Yes, now LAC I gene produces the repressor protein. Yes, so whenever glucose is present, what happens? Whenever glucose is present, there is decreased availability of adenylyl cyclase. Okay, so if adenylyl cyclase is less, what happens? Cyclic AMP levels are less. So the cyclic AMP levels are less. The cap protein cannot be bound to cyclic AMP. There is no cyclic AMP to bind to the cap protein. So here the cap site cannot be bound with the cap and the cyclic AMP protein. So here the cap site is empty. Now the repressor, it is present, but inducer is not present to block this repressor. So what happens? The repressor can go and bind to the operator site. So the operator site is now occupied by the repressor. So what happens? The RNA polymerase cannot initiate the transcription. Yes, so thus the lactose operon is in the off condition. So the transcription cannot happen. So the glucose is being utilized by the bacteria, right? So whenever glucose is present, the LAC operon is off. How? There is reduced cyclic AMP levels. So cyclic AMP cap does not bind to the cap site. Also the repressor which is formed binds to the operator site and thus blocks the transcription of the genes. So this is what happens whenever glucose is present. So the next scenario is when glucose is absent and lactose is present. So in this condition, the bacteria has to utilize the lactose. So what is this? It is the LAC gene which produces the repressor protein. Yes, this is the cap site. The cap site where the cap and cyclic AMP binds. And this is the third one is this is the promoter site, and this is the RNA polymerase, and the operator site. Yes, and next are three genes, the genes which code for beta galactosidase, permease, and thiogalactoside transacetylase. Yes, so now what happens? Glucose is absent. Yes, glucose is absent. So the cyclic AMP levels are more. So the more cyclic AMP levels can bind to the cap protein. So the cyclic AMP plus cap protein can go and bind to the cap site. Yes. So this is one factor which can favor the LAC operon. The next one is the repressor is produced. So what will lactose form? The lactose present, some of the lactose can form allolactose. Yes, allolactose is an inducer that can bind to the repressor protein and it can block the action of the repressor. So the repressor will not go and bind to the operator. So now our operator site is free. Yes, now the operator site is free. So now the RNA polymerase can initiate the transcription of the three genes, and the three enzymes can be involved in the lactose metabolism, and now lactose can be utilized by the bacteria. Yes, so this is where you call the LAC operon to be in the on condition. So what happens when lactose is present? The cyclic AMP levels are high, that can bind to the cap protein. So together it can bind to the cap site, and whichever repressor is being formed is inhibited by the presence of an inducer which is produced by the lactose. So the repressor cannot bind to the operator site. So the operator site is free now. So the RNA polymerase can initiate transcription and the three enzymes can be produced for the lactose metabolism to occur. Yes, so this is one scenario where the lactose is present. So the lactose operon is in on condition. The third scenario is when both the fuels are present, that is glucose is also present, lactose is also present. So what is, again let us write, this is LAC gene which produces the repressor. I am repeating it because you have to memorize this, okay. And the next one is the cap site where our cap and cyclic AMP binds. This is the RNA polymerase which initiates transcription, and this is the promoter site. Next is the operator site, and these are the three genes which are involved in the lactose metabolism. So glucose is present. So if glucose is present, yes, if glucose is present, then our cyclic AMP levels are less. Yes, so there are no cyclic AMP to bind to the cap site, so it cannot bind to the cap site. Yes, there is no cyclic AMP to bind to the cap protein, so that cannot bind to the cap site. Now the repressor which is found can either go and block the operator site and inhibit the transcription of genes, or there is presence of lactose, right? So some amount of lactose can produce allolactose which can inhibit the repressor. Okay, so some of the repressor protein can go and bind to the operator site and block the transcription. Some of the repressor proteins are blocked by the presence of an inducer, allolactose, which is produced from the lactose. And also we know that since glucose is present, the cyclic AMP levels are less. So cap site is now not bound to the cap protein, cap plus cyclic AMP protein. So what happens in this kind of a condition? few mRNAs are produced. Okay, few genes are being expressed. Few genes are expressed. So the LAC operon is not completely inhibited, but it is not induced. Okay, LAC operon is uninduced. So few genes are expressed, right? So this is the condition where the LAC operon is uninduced. So we have three scenarios. We have seen three scenarios wherein if LAC operon is off whenever glucose is present, and LAC operon is on whenever lactose is present. Yes, and LAC operon is uninduced whenever both glucose and lactose are present. So this is about the LAC operon concept.